Hi guys, today we're solving Cambridge A-Level Biology Paper 1 Topicals, Biological Molecules, Chapter 2. What is the general formula for amylose? Amylose, the monomer for it is alpha glucose. The chemical formula would be C6H12O6, but remember it is joined together to form glycosidic bonds by condensation reaction, and in condensation reaction a water molecule is lost, so you would have C6H10O5. So even if you don't know, you can figure it out like this, and our answer is D. Question number 2. Which statement describes how the molecular structure of starch is suited to its function, how it's suited to its function? So there could be other points that could be accurate, but they may not be related to the function, so you have to be careful. A, amylose has a branch structure and amylopectin is coiled to give a compact molecule for transport. Now that would be wrong because it's the other way around. Amylose is coiled and this is branched. So this is not our answer. B, in the breakdown of amylose and amylopectin, many condensation reactions release stored energy. Okay, but is that related to the function? Not really. In the formation of amylose and amylopectin, many hydrolysis reaction allow the release of stored energy. So it says in the formation of it, there are hydrolysis reaction. No, in the formation it's condensation reaction, and in the breakdown it would be hydrolysis. So that's wrong. And then D is our answer. Let's see what it says. Amylose and amylopectin forms a complex that is insoluble and so does not affect the water potential of the cell. So that's relating to how it's suitable for its function. What's that function? Its function is storage. The structure of phospholipid includes a polar phosphate head, hydrophobic fatty acid chain, saturated fatty acid chain. What are essential for the formation of phospholipid bilayer in a cell surface membrane? So remember, phospholipid, it's going to have a phosphate head that is hydrophilic, and then it's going to have a hydrophobic region that is fatty acids. It's going to have two fatty acids and it's going to have a glycerol in the middle. Polar phosphate head, yes. Hydrophobic fatty acid chains, yes. But saturated fatty acid chains, that is not really the case since it's both saturated and unsaturated. So it's 1 and 2A. Question number 5. The diagram shows the molecular structure of a peptide. It's a peptide, so it's made up of amino acids. And let me draw it. So typically you would have a carbon atom in the center, and it's gonna have 4 bonds. You have R group, you have a hydrogen here, and then you have the amine group, and then you have another carbon here for COOH. You can write it this way as well. So this is how you'd have your amino acid. So over here it's a peptide, so there's a few amino acids joined together. So if I were to break them down, it would be one here, another one here. So those are 3 amino acids. Which molecule would result from a complete hydrolysis of the peptide? Hydrolysis is breakdown. This is breakdown. And what happens in hydrolysis is that you have an addition of water molecule. at each point where the hydrolysis takes place. So let's look at it. For the option A, as you can see, this is the R group, this is the R group for the first amino acid. This is the first, second, and third amino acid. For the first one, this whole part would be the R group, but over here, does it match? No, it doesn't, because the CH2 is appearing twice. So this is wrong. So A is not our option answer, and then B, if you look closely at the second amino acid, it's this bit is the R group, and the R group here does not match with that, so this is not our answer. The third option, everything looks right, so this is an answer. You can see each R groups are the same and everything else is correct. And then D is wrong, because why is D wrong? The third one has H on both ends, whereas the R group is supposed to be CH3. So that's where it's wrong. So C is our answer for this. Question number 6, the diagram shows a hemoglobin molecule. Which row identifies the different parts of the molecule correctly? So what is A? A is, it's pointing towards the prosthetic group, but since it's hemoglobin, you know the prosthetic group for hemoglobin is going to be the heme group. So it's the heme group, and that is also the oxygen binding site, so it could be either of these. The next one, 2, that is the outer region, so hemoglobin is globular like this, and this is the outer region. It's facing outwards. So is it going to be hydrophobic or hydrophilic? It's outwards, facing outwards. So it's going to be the hydrophilic amino acids, not the hydrophobic ones. Number 3 is here. That would be your coiled, that's the alpha, see it's a coil, so it's alpha helix structure. And then you have 4, that's pointing inwards, so in the center you're going to have the hydrophobic amino acids. So our answer is B. Question number 7, which color indicates the lowest concentration of reducing sugar present in a solution after testing with Benedict's solution? So the key point here is lowest concentration. So it goes from blue to, in the end you get a brick red color if there is a high concentration, but otherwise you would get orange, yellow, and green. So it's easier to remember it like the rainbow colors, red, orange, yellow, green, blue. So coming backwards, the lowest concentration is here from blue to green, so it's B. The diagram shows a monosaccharide xylose, a monosaccharide. Many xylose monomers can be joined by glycosidic bonds to form a polysaccharide, which is found in plant cell walls. Which diagram shows the formation of a glycosidic bond between 2 xylose monomers? So this is a condensation reaction, formation of glycosidic bonds between 2 of them, between 2 monomers. So this is going to be, let me write it down here, condensation reaction. And in condensation reaction, a water molecule would be lost, and you'd have the oxygen joining the carbon of both monomers. So let's look at our options here. A is wrong because it's supposed to lose a water molecule, so H2O. It's only losing 2 hydrogens. So this is wrong. In this case, it's losing the whole thing. H2O is lost, but you don't have any other extra oxygen to, there is no oxygen to join the 2 carbons. There's supposed to be an oxygen remaining for that bond to form. Then the third one, you're losing both hydrogens, so this is not even right. And the last one is right because you lose H2O, lost, and in the end you have this oxygen that's going to join the 2 carbon together. Next question. Question 9. Which molecules are monomers? So monomers. Ribose, it is a monomer, yes, because it is a 5-carbon sugar. It is in RNA nucleotide structure. Ribose is found in there. Then glucose is also a monomer. Remember, glucose and a glucose forms a maltose. You have glucose as a monomer of starch, of cellulose. Deoxyribose, this is similar to ribose but in DNA instead of RNA. Again, this is also a 5-carbon sugar monomer. The difference between ribose and deoxyribose is that it has one less oxygen compared to ribose than ribose. Just so you know, for that kind of question if it comes up. Sucrose is not a monomer because sucrose is made up of glucose plus fructose, which are 2 different monomers. So our answer is A. Question number 10. Which molecules have properties that are dependent on hydrogen bonds? So dependent on hydrogen bonds only, that would be cellulose. Does it have hydrogen bonds, and is it dependent, are the properties of it dependent on it? So cellulose definitely, because it has hydrogen bonds form between adjacent beta glucose chains, and this provides the structural support that it needs. Structural support in the plant cell wall. The next one, glycogen, is no, because it is highly branched. That is one thing. It is highly branched, and its properties are like solubility and let me just write it down. Properties like solubility and how compact it is, that is not really dependent on hydrogen bonds. Solubility and compactness dependent on glycosidic bonds and not hydrogen bonds. The next one, hemoglobin. Yes, because hemoglobin, it is a globular protein, and the secondary and the quaternary structure is stabilized by these hydrogen bonds. Stabilized by hydrogen bonds. Water is dependent because properties of water like high specific heat capacity, high boiling point, surface tension, all of those are due to the hydrogen bondings. High boiling point, high surface tension. Those are all dependent on the hydrogen bonds. So it's 1, 3, and 4. Our answer is C. Some features of triglycerides are listed. By the way, triglyceride is going to have a glycerol and 3 fatty acid chains. 3 fatty acid chains and a glycerol molecule. Some features of triglycerides are listed. Which of these features make triglycerides suitable energy stores? So all of them are true, but we have to relate it to which ones are good, suitable energy stores. So it's going to be the fact that it can be liquid or solid at room temperature. That's not really related to solubility and not really related to energy store, because it's just a physical state, so it's irrelevant. It contains a high proportion of carbon-hydrogen atoms, so yeah, because this is where energy is stored. So this is energy storing. Carbon-hydrogen bonds are important. Insoluble in water, that is obviously directly related to how it's a good energy store, because then it would not be affected by osmotic potential. Less dense than water, that's not related to this, because this is more of related to buoyancy. So D is our answer. Next question is question number 12. Some foods contain hydrogenated vegetable oils. These are unsaturated fats that have been converted to saturated fats. So what do we know about these? Unsaturated and saturated fats. Unsaturated are going to have carbon-carbon double bonds, and they're going to have kinks. And saturated bonds are carbon-carbon single bonds. So they have been converted from this to this. Which property of the fats will have changed? So we'll see which one changes because of this. Their hydrocarbon chains will fit together more closely. This is our answer, because when it goes from double bond to single bond, we get rid of the kinks. So it removes the double bond through this process of conversion. So we have A as our answer. B is their solubility in water will increase. No, because it does not affect solubility. And they will have more double bonds in their molecules. No, because they'll have less double bonds now that we have converted from unsaturated to saturated. They will remain liquid at room temperature. Remain liquid, that would be wrong, because they will actually go from liquid to solid, because they're going from unsaturated to saturated. 13. Which statements could be used to describe enzyme molecules and antibody molecules? So remember, enzyme molecules, they are 3D shaped molecules. Enzymes, you know what they are, both are protein. Antibody molecules are Y-shaped immune system proteins. 1. Hydrogen bonds stabilize the structure of the protein and are important for it to function efficiently. That is true. Hydrophilic R groups point to the center of the molecule and cause it to curl into a spherical shape. So hydrophilic will never point to the center. Hydrophilic are on the outside facing outwards, and it's the hydrophobic that would be inside pointing at the center. Number 3, the tertiary structure of the protein molecule plays an important role in the function of the protein. You know that because as the tertiary structure changes, the 3D shape changes, and then the function is going to be affected by that. So it's 1 and 3. C is our answer. Question number 11. A student carries out 4 tests for biological molecules in a solution. The results are shown in the table. So these are the tests carried out, and this is our result, the observation. Is, in this case, iodine, that is going to be for starch, and its observation is orange-brown. So you know there is no starch, or starch is absent. This one, if this is the test for protein, you know, it's supposed to go from blue to purple. And since it's purple, that means protein is present. The next one, Benedict's, that is for reducing sugars. So it turns orange, that means it's red sugar, reducing sugar. Reducing sugar present. Emulsion test, that's for lipids, fats. This is still clear, so there are no fats or lipids. But if it was milky, cloudy, then that would be present. Now let's see which one of these would be present in the solution. Glucose, starch, globin. Starch is not present. We saw that here. B, globin, glucose, and collagen. All 3 are present. Globin is globular protein. Glucose is present because reducing sugar was present. It was orange. And then collagen is for protein. And then C, starch is not present, so this would be wrong. B is our answer, but D is sucrose, globin, and glucose. This is sucrose. Sucrose was not present because we only conducted the test for reducing sugars. Next question. A student was asked to estimate the concentration of reducing sugar in an unknown solution using the Benedict's test. 5 reducing sugar solutions with different concentrations were provided in order to produce a calibration curve. A student added 2 centimeter cube of Benedict's solution to each of the reducing sugar solutions, heated them in a water bath, and recorded time taken for the first appearance of a color change. Which variable should the student standardize, so keep it constant, when carrying out the Benedict's test on each reducing sugar solution to ensure the results are comparable? So this is concentration of reducing sugar in an unknown solution using Benedict's test, and they added 2 centimeter cube of Benedict's solution to each, and heated, and then recorded the time taken for the first appearance of color change. Which variable should they standardize? Standardize keeping constant, keep it same to ensure comparable results. Volume of reducing sugar used, that should be kept the same. The temperature of water bath, that should be kept the same. The time the solutions are heated, remember this bit, it says they have to record the time for the first color change to appear. So that is going to vary. This varies, so this you cannot keep the same. So it's 1 and 2. Answer is B.